Welcome back, student, to part 2 of chapter Locomotion and Movement. Let's start. In this video, we will discuss the structure of striated muscle. Skeletal muscle is made up of many muscle bundles which are known as fascicles. Fascicles are held together by a collagenous connective tissue layer which is known as fascia, and each fascicle contains many muscle fibers. Muscle fibers are lined by the plasma membrane, and that is known as sarcolemma, which encloses sarcoplasm. Each muscle fiber contains parallelly arranged myofilaments, which are known as myofibrils. Each myofibril has alternate dark and the light striation. Dark striation is known as anisotropic or A band, whereas light striation are known as isotropic or I band. Now, why it is so? This is due to the presence of two fibrous contractile proteins, thin actin filament, and the other one thick myosin filament. I band contain actin filament and the A band contain actin as well as myosin filament. They are arranged parallel to each other. A band bears lighter middle region which is known as H band or the H zone. Here you can see in the figure, and it is formed of only myosin. A thin dark line which is M line runs through the center of H zone. I band is bisected by a dense dark band called the Z line. Region between two Z line is known as sarcomere. Sarcomeres are the structural and functional unit of muscles. Here you can see one more diagram of muscle, skeletal muscle. We can see over here the Z line, H zone. Between the H zone, there is M line. Then dark band is the A band and the lighter one is the I band. Let's see the structure of actin, that is a contractile protein which is present in skeletal muscle. An actin filament is made up of two filamentous actins which form double helix. So it is F actin which is filamentous actins. F actin is a polymer of monomeric globular G actins. So here you can see the circle loop are the G actins which are globular in structure. Actin contains two other proteins which are known as tropomyosin and troponin. So it has three basic structures: F actin, tropomyosin and troponin. Two filaments of tropomyosin runs along the grooves of the F-actin double helix. Whereas the troponin has three subunits, it is located at regular intervals on tropomyosin. In the resting state, a subunit of troponin masks the binding site for myosin on the actin filament. That makes the actin filament at the resting state, which will not allow actin filament to bind with the myosin. Now let's learn the structure of myosin. Each myosin filament is polymer of many monomeric proteins which are known as meromyosins. A meromyosin has two parts: heavy meromyosin, which is HMM, or the cross arm, which is a globular part, head plus the short arm, and it projects outwards. Whereas the light meromyosin other part is the LMM, which is the tail part, or the light meromyosin. The globular head is an active ATPase enzyme. Here you can see over here the globular part. It has actin binding site. Also, it shows the ATP binding sites because it has an active ATPase enzymes to it. It has binding sites for ATP and active sites for actin. So myosin binds with the actin with the help of ATP molecule. Now we will learn the mechanism of muscle contraction. According to the sliding filament theory, contraction of muscle fibers occurs by sliding of the thin filament, that is actin, over the thick filament, which is myosin. Now these are the steps involved for contraction, muscle contraction. The first step is an impulse from the CNS, that is central nervous system, reaches to the neuromuscular junction, which is the motor end plate via motor neuron. Neuromuscular junction is a synapse between a motor neuron and the sarcolemma of muscle fiber. A motor neuron plus muscle fiber is equals to a motor unit. The second step after that is synaptic vesicles will release the neurotransmitter, which are acetylcholine. Then it generates an action potential in the sarcolemma that spreads through the muscle fibers. This will cause release of calcium ions in the sarcoplasmic cisternae into sarcoplasm. That will cause increase in calcium ions. This calcium will bind with a subunit of troponin on action filaments and unmask the active sites of myosin. Here you can see that calcium is binding to the troponin. Now that will unmask the active sites for myosin to bind with actin. Using the energy of ATP, hydrolysis, myosin head will bind to the active site on actin to form cross bridge. You can see over here the cross bridge is formed. This will pull the actin filament on both sides towards the center of A band. Now the actin filaments partially overlap so that H zone will disappear. Now because of the pulling of actin, H zone will disappear. Next, the Z line attached to the actin is also pulled inwards. You can see over here in this diagram the contracted and the relaxed muscle. The Z line which is attached to the actin is also pulled inwards. This will cause the shortening of sarcomere, that is contraction of sarcomere. Here the I band gets shortened and A band will retain its own length. There is no change in the A band. Myosin will release ADP and PI and goes back to its relaxed state. A new ATP binds and the cross bridge is broken. The ATP is again hydrolyzed by the myosin head and the above process are repeated, which will cause further sliding. Now here you can see in the diagram the relaxed muscle where H zone is visible, I zone and A band is visible. In the contracting stage, as H zone, I zone will reduce, whereas there is no change in the A band. When the maximum contraction takes place, H zone and the I zone disappears. It's same A band, there is no change in the A band. When calcium ions are pumped back to the sarcoplasmic cisternae, the action filaments are again masked. When the calcium filaments are not available, that will lead to masking of again the myosin site of the actin filament. As a result, the Z line will return to their original position and this results in relaxation of muscle. The reaction time of the fiber varies in different muscles. It's not same in all the muscles. All the muscle doesn't show the contraction at the same time or relaxation at the same time. It varies from muscle to muscle. Repeated activation of the muscle leads to accumulation of lactic acid and that causes muscles fatigue. This is due to anaerobic breakdown of glycogen in muscles. Now, on the basis of the presence of myoglobin in the muscles, skeletal muscles are of two types, red muscles and the white muscles. Myoglobin, let us first learn what is myoglobin. Myoglobin is this pigment, muscle pigment. It's a iron containing muscle pigment. It's same like the hemoglobin which is present in RBC. Now let's learn the difference between aerobic muscle and the anaerobic muscle. That is white muscle fiber and the Red muscle fiber. Red muscle fiber, it is red in color due to more amount of myoglobin. White color due to the lesser amount of myoglobin. Red muscle fiber have more mitochondria, they show aerobic metabolism, and their contraction is slow but sustained. Whereas on the other hand, white muscle fibers, mitochondria is less, they perform anaerobic metabolism, that's why they are also known as anaerobic muscle fibers, and the contraction is fast but for the shorter period. So this is how the white and the red muscle fibers are differentiated on the basis of their function, metabolism, and because of the presence of pigment. Thanks for your attention.